Arnold Mata will be played today by Emily Martin. That's me. And this is Lydia Mendoza, who will be co-moderating the session with me. And so, probably need a second to get slides ready.
Yeah.
We'll commence in just a moment. So while getting set up, I'd like to introduce Shidan Tosif, who will be presenting the talk Southern Hemisphere Cross-CEIRR Collaboration, Natural History of SARS-CoV-2 in Comparison to Influenza A in Australian Perspective.
Good morning, everyone. Hopefully you can see my slides. And yes, it's really great to be here and present on behalf of our Southern Hemisphere Cross-CEIRR Collaboration. My name is Shidan. I'm a pediatrician at the Royal Children's Hospital in Melbourne and also a researcher at MCRI. And many of us involved in this project were new to CEIRR, so it's been really great to see all the wonderful activity through this program, and I've got a lot to take back to our team in Australia from these wonderful presentations over the last few days. Just to briefly introduce our team, we have representatives across 3 of the states in Australia who've contributed to our study. And I just wanted to highlight our Pediatric Active Enhanced Disease Surveillance Program, the PEDS network, that also contributed a lot of data to this study. They are a team that looks at prospective case ascertainment for important and serious childhood diseases in Australia, and influenza is one of our surveillance diseases. So my aim here really is to try and describe and characterize the natural history of the respiratory viral diseases that we've seen in Australia over the last 2 years, focusing on COVID, influenza, and RSV. And I'll be drawing on a combination of hospitalization data, surveillance data, and also trying to share some of the healthcare impact which we've experienced, particularly over the last few months. And just to provide some context to the data that I'll be presenting, as you've probably heard, the case numbers for COVID in Australia were quite low throughout most of the period, so we actually didn't have much to describe until this year. And even more so, we didn't have other viruses circulating, so therefore, much of the activity that I will share is from the last few months. And therefore, it's a little bit raw, some of the data. Our team tried their best to pull together as much as we could for this presentation, but hopefully you'll see some of the common themes emerging. And also, due to the hard border lockdowns, we saw a lot of differences between states in Australia, including particularly from Western Australia, where there was very little community transmission of viruses until March of this year. And that led us to suspect, as we have for a long time, that Western Australia always wanted to pull away from the rest of Australia, and their policies towards COVID really highlighted that. One of the early strengths I think we could reflect on is that we had an establishment of a COVID-19 platform, and this has supported a breadth of studies through predominantly a large household transmission cohort that has now over 500 cases and close contacts. We have biobank samples of PBMCs, serum, plasma, granulocytes at multiple time points. And I think one of the wonderful things about this platform has been the ability to collaborate with groups such as Katrin Ketsierska's lab and Tanta Subaro's lab, which has been a real highlight for us. So now just moving on to our experience with COVID, and I will just try and focus on the last 6 months where most of the action has been for these viruses. You can see that Australia took an aggressive suppression approach where through lockdowns and social restrictions, much of COVID activity was suppressed until late 2021. And the aim was to improve vaccine introduction and adherence. And that was where, following improved vaccine adherence, we had our release of restrictions coinciding with the Omicron waves earlier this year. And that's now resulted in kind of a bit of a steady state of COVID infection since that time. Looking at Australia's profile for COVID, this reflects a similar experience to many other countries whereby the cases are distributed with a significant proportion of cases in the younger childhood and adolescent age groups. However, deaths are very much overrepresented in the elderly and older age groups. And I just wanted to highlight our vaccination profile for COVID in our state, which we have a very good uptake in the older age categories. And our uptake for the 5 to 12-year-olds since being introduced in February this year is close to 50%. And as of next month, we will be introducing the vaccines down to 6 months of age. And we've published a number of papers describing the profile of COVID in children in Australia, and I won't go into detail into these at this time, but I really wanted to focus on what we've seen and compare across the three viruses over the last 6 months in particular. And so, looking at our hospital level data, since the beginning of this year, we have had just over 600 admissions. Most, as you can see, are in children under the age of 5 years, with clinical features similar to most other respiratory viruses, predominantly bronchiolitis and viral symptoms in the older children. Notably, most of the children that have been admitted have not required any medical interventions. These are typically children who were admitted for observation for fluids and oxygen support, but didn't require those interventions. And we had to look back across the length of stay, finding that our length of stay has reduced with Omicron. And notably, our intensive care and invasive respiratory support requirements have also reduced in the period of Omicron as compared with Delta. However, there's a real clear signal of croup, and that's been described in the United States as well with Omicron. And another clear standout is the number of children we're seeing now admitted to hospital are really overrepresented in terms of those children who have comorbidities. We've had very few deaths from COVID in children in Australia, and we've had one this year, sadly, with a child who died with COVID rather than specifically from COVID. And I just wanted to flag that thinking about the impact of COVID, it's important to reflect on PIMS-TS, or as it's called in the US, MIS-C, multi-system inflammatory syndrome in children, noting that we've had 22 cases with 6 requiring intensive care in Australia this year. In Victoria this year. When we look at our hospital admission data, the timeline really reflects that those periods for us which have been busy are those periods of high community transmission. However, reflecting on how much of a burden this is on our hospital systems, this has not been a major feature in terms of the impact for us. Weekly, at most, there was 40 admissions per week at our hospital. And as you'll see, compared to other viruses, this is not a large number. And just to flag, you can see on the right side of this chart here, the number of croup. I just wanted to flag how much of that has really been contributed by COVID, particularly this year. And when we looked at the vaccination status of our hospitalizations, we found that most children were not vaccinated that required hospitalization. Around about 2/3 of those between 5 to 11 years of age were not vaccinated, and about 1/3 to 2/3 of those who are in the 12 to 18-year-old groups were not vaccinated who were eligible. And noting again that most of those older children had comorbidities, most would have risk factors that place them at high risk of needing hospitalization anyway. But at the same time, the vaccines becoming available for us coincided with when we had our Omicron surge at the same time. So there's probably a bit of overlap with what was available vaccination-wise and also those infectious exposures. Now moving on to influenza, and you've probably heard that we've had a very significant and rapid flu season this year in Australia. And you can see in the red line here just how prominent our 2022 flu season has been as compared to previous years prior to the pandemic in 2019, 2018, where typically we have a season which comes on later in winter. We had a very early season this year and a significant suppression over the 2020 and 2021 years. Our vaccination rates at the middle of this year stood around 40%, for all people over the age of 6 months. And it's worth noting that in Australia, the vaccine for flu is funded for children under the age of 5 and those who have special risk factors. But whilst it's recommended, it's not on our formal national immunization program, so it does require parents and caregivers to really take up the vaccine. And interestingly, when we look at the 6 month to 5 year influenza vaccine coverage rates, we can see here in 2020 a higher vaccination rate for flu in this age group as compared to this year and last year. So we've kind of lost a little bit of momentum with flu vaccination in this higher risk group. And many of the families we speak to have really got COVID at the front of their mind and COVID vaccination. And I think that's replaced a little bit of the attention to the flu vaccine, which may be a reason underlying this difference. We've also seen in Australia a very high or a higher proportion of influenza, particularly in the younger age groups. In 2022, people aged 5 to 9 years, and those younger than 5 years represented those with the highest notification rates. And you can see the kind of balance of this graph in 2022 reflecting that as compared to previous years where there's a bit more of an even distribution and the older and the younger age groups were a bit more balanced as opposed to what we see this year. And again, moving to the hospital level data, we can see we had our surge in admissions for flu, particularly over the April, May, June months, reflecting community transmission. And whilst we didn't have as many numbers compared to COVID admissions this year, these admissions for flu were really concentrated over a short 2 to 3 month period, so it felt a lot more intense for us as opposed to COVID. Much of the age distribution reflected the younger age groups in 5 to 11 years, with a clinical phenotype that we're all familiar with flu. But at the same time, we saw a 10% roughly admission rate into our intensive care unit with problems such as encephalitis. We had severe bacterial complications, sepsis, and sadly there were 2 deaths as a result of flu complications. And again, looking at the flu vaccination status of children admitted to hospital, very few children have been vaccinated who are hospitalized, less than 10%. So again, this really stands out in terms of the engagement with the flu vaccine this year. In terms of the subtypes we've seen of flu, most have been in the H3N2 and H1N1 this year. And I'll finally just touch on RSV because we have a very nice WHO SARI surveillance program, which includes 4 pediatric hospitals across the country. And each of these sites has its own unique demography, with Darwin in the northern hemisphere having a tropical monsoonal climate, where RSV is traditionally linked with the rainy season, as compared to other parts of Australia where we have a more typical winter pattern for the RSV season. And this is a year-round surveillance program with sequencing completed by the WHO Collaborating Centre Lab with Professor Ian Barr. And again, we've had unseasonal RSV peaks. As you can see, in 2021, we had a very uncharacteristic peak in late summer for us, which impacted almost all jurisdictions apart from WA. And this year, we've returned to a more typical pattern with regards to the RSV occurring in winter. However, what's really been clear is the sheer number of cases which we've had in the last 2 months has really outstripped anything we've seen in previous years. And again, looking at hospital data, we've had a surge of RSV, and this has probably had the most significant healthcare impact in terms of demand for hospital level care, but also in our community. And it's really impacted access in the hospital more broadly. For example, our intensive care beds have been all filled with patients with viral infections, and the flow-on impact of that for elective surgeries after a prolonged pandemic period has really been significant. And when you look at the viral trends side by side, you can see that basically in the last 6 months, or since the beginning of this year, we've gone from a COVID wave to flu to RSV, moving almost sequentially from one to the other. And not to forget that there are the other viruses that have really come back into circulation as well, such as enterovirus, rhinovirus, all in the mix and contributing to our experience. We went back and looked at co-detections, and interestingly, we had quite a number of flu and COVID, flu RSV, and some even with flu, RSV, and COVID. However, it wasn't really a feature in terms of those children needing hospitalization, but these are small numbers. However, those that had a bacterial complication obviously had more significant morbidity. And this is not from our group, but I just thought it was of interest from George and colleagues in Australia who looked at the rates of influenza and invasive meningococcal disease, where in the past they've often found flu and rates of meningococcal disease being related, and again noting that they've seen suppression of meningococcal disease during the pandemic, and we just wonder, what are we going to see as well as what we've seen with respiratory viruses? Is there something yet to be seen with other diseases such as meningococcal disease? But we really haven't picked up a signal for that yet. And finally, as I've alluded to, these combination of viral peaks, successive viral waves, together with what has really been high rates of respiratory virus in our staff and their family members impacting their ability to work. We still have COVID isolation measures, which impact staff availability, as well as a significant degree of fatigue and burnout from the pandemic period, has led to major disruptions in our hospital systems. Usually we anticipate a significant winter, but all these additional factors have led to a large backlog of our medical and surgical cases, and the thought is that this is actually going to take quite a few years to overcome as a result of what we've seen this year. So in conclusion, we've seen a major rebound of respiratory viruses compounded by sluggish vaccination rates, particularly for influenza. We feel that children, and we may be biased as pediatricians, but children have been disproportionately impacted through the pandemic by the direct and indirect impacts. But at the same time, this year, the significant number of respiratory viruses circulating, particularly in children, has impacted again their well-being and their school attendance, as well as being unwell. We wonder how long we're going to see this kind of immunity debt that we've accumulated over the last few years play out. And I think what we've observed has implications for vaccine approach and policy considerations, and we really need to revitalize education and support for influenza vaccine. Our experience has been, in terms of gross numbers, that RSV has really had the most impact in terms of hospitalization if you look at numbers and the requirement for health service delivery. However, in terms of disease severity, influenza certainly has had the most significant impact in terms of disease severity and hospitalization, followed by COVID. But I think COVID we've taken into account PIMS, TS, and MIS-C when we factor in disease severity. And as I've touched on, the significant secondary health impacts. So with that, I just would like to acknowledge our wonderful team that was involved in this study, and really thank CEIRR for the opportunity to be part of this. Hopefully this is a first of many more collaborations, and I'd love to be in touch and hear from anyone else who'd like to collaborate. So thank you for your time.
So we have a few moments for questions.
Thank you very much. Thank you for two things. One, to talk about children as a pediatrician, and second, to talk about RSV, because sometimes the people think that it's not very important. No, I'm amazed your experience is totally different than the one that we have. I work in Columbus, Ohio, because 60 to 70% of the admissions, we have 3,000 admissions with COVID, were teenagers, and 80% require treatment with remdesivir and dexamethasone. So your teenagers must be something much better than ours. One aspect that I want to ask you, do you have a lot of obesity among teenagers? Because the teenagers that require more severe interventions were obese. Do you have that in your part of Australia?
Yeah, thank you. That's a great point, and I think reflects the real narrow window of the data that I presented. Because if you look at what happened last year for us, certainly those children and adolescents with obesity were more impacted. I think our obesity rates are not as high as the United States, definitely, but still that was a clear risk factor for us, and needing the kind of more intensive treatments as you've described. I think what's helped this year is that we've had really good vaccination rates, particularly in those children over the age of 12. It's approaching over 90%. And so that, together with the difference in strains impacting what we've seen, I think is that difference. But certainly last year we had many adolescents represented.
Because, since 2004, when we have the big pediatric outbreak in flu and we realized that obesity was such a high risk factor, it has been always consistent. We always get teenagers with flu as well. Now, if I may, about the RSV, my favorite virus, you saw a little bit of co-infections. We saw 50 or something, 5%. That when there was Delta and RSV last summer, we have not seen the surge yet of RSV yet. Because as you said, our ICUs are full, the ED is full. But half of our kids end up in the ICU when they have COVID and RSV, but that has not been your experience. Totally different. I'm really amazed how the opposite is what we have seen, right? So,
yeah, I agree, and we would have thought children who have either successive or co-infection are more at risk, and that's why we went and specifically looked back at that. And I think it needs to be unpacked a little bit further. And some of these differences in what we're seeing across different settings would be interesting to unpack. Because there must be some basis for this, why we're not seeing that in our setting.
You're working in the context of Omicron, right? Not
I just presented Omicron.
Right, because that was Delta more. So we'll see what happens if that happens. But thank you very much. It was a
pleasure.
So I think that it's time to invite our next speaker to the stage. I have a question for you, but I'll ask you after this session. So thank you very much, Dr. Gunn.
All right, I'd like to introduce Kendall Witt, who is going to be presenting on the natural history of SARS-CoV-2 infection in Colombian populations.
All right, so you guys can see my slides? Okay, all right, so thank you. My name is Kendall Witt. I'm a second-year graduate student in Dr. Stacy Fulcher's lab at St. Jude, and today I'll be talking about SARS-CoV-2 in Colombian populations. Slides aren't progressing for you guys either, are they? No? Okay. All right. So I don't have any pull up on computers. Okay. All right. All right. Okay. There we go. Now we're good. All right. Thank you. All right. So as we all know, we are now in the third year of the pandemic, and COVID is still here and still going, unfortunately, pretty strong. We have seen almost 600 million infections and over 6 million deaths. We have seen multiple variants and subvariants that have emerged during this time. At the end of 2020, early 2021, we did see the deployment of vaccinations, and at this time we've seen over 12 million vaccinations deployed in over 200 countries. And so with the general spread of the virus and immunity built from these infections as well as from these vaccinations, we have seen a decrease in the daily infections and deaths. So as we have learned from this talk and just from general news reports, we're certainly not out of the weeds yet. Since the emergence of the virus and all throughout the pandemic, and especially with the emergence of these variants and also vaccinations, a lot of studies have focused on what the general immunity to SARS-CoV-2 looks like. What is the longevity, what is the breadth, and especially with the vaccinations and variants, how protective are we based off of the type of vaccines that we receive and what variants we are exposed to. A lot of these studies do typically focus on Western populations, and for the vaccination studies, a lot of them focus just on the mRNA vaccines. And so thanks to our collaborators in Colombia, we have been able to look at some of these questions in a northern Colombian cohort. So the goals of our Santa Marta COVID study is to understand how the immunological responses differ by ancestry, race, sex, age, and strain exposure, evaluating the serological versus mucosal protection, looking at the roles of coviral and coparasitic infections, and then also looking at the influence of vaccinations on immunological responses, particularly how different vaccine platforms can have different influences, whether partial vaccination is enough, or if you really need to have that complete vaccination. And finally, what is the influence of boosters? Do they tend to skew towards one variant versus another? To do an overview of our cohorts, majority of our individuals are from a northwestern town, rural community called Santa Marta. So we do have some indigenous individuals that are a little bit further inland from El Limon, the Wiwas. In order to be enrolled in this study, individuals must be infected with SARS-CoV-2, and this is confirmed via either PCR or rapid antigen test. And we do have rolling enrollment. Enrollment began in September 2020, though we are still accepting individuals into the study, with some new enrollees as of April and maybe even more recently since then. For sample collections, we collect blood and nasopharyngeal swabs on day 1, 30, 90, 180, and day 365. To look at the demographics of our cohorts, we have a total of 114 enrollees at the time for this analysis, so we do have a few more now, and we'll hopefully get those samples soon and be able to add them to the analysis. The median age is 41 years old, with an age range of 14 to 77 years old. We do have slightly more females than males in the study, with about a 60-40 split. And we do have over 50% of them that have been vaccinated, though as I'll mention in a few slides, we do not have the post-vaccination samples for all of those individuals yet, though we will hopefully have them soon. Luckily for majority of the individuals within our cohort, most of them only had mild disease. Many of them suffered or reported symptoms of cough, sore throat, headaches, and general malaise, but none of them had to be hospitalized or put on ventilation. Some did continue to report symptoms all throughout the study. The majority of them just experiences major symptoms during their acute infection. As I said, we have a rolling enrollment, with enrollment starting in September 2020. And for the purpose of this talk, we have sample collections for individuals that were enrolled as recently as February 2022. The majority of the individuals were enrolled in April through July 2021, which, looking at the variant spread map of Colombia at that time, we see that that is whenever mu was the main variant circulating at that time point. Comparing to the US and other countries throughout the world, the variant spread is very different. Mu is not able to really get a strong foothold outside of Colombia. And so comparing this to other cohorts will allow us to really be able to hopefully tease out how that strain exposure can influence the immune responses. To be able to analyze our cohort, we divide them into 3 groups. Our first group is the infection-only group. So these are individuals who were not vaccinated, or as you see from the little asterisk, some of them have been vaccinated, but we have not received any of those post-vaccination samples. And once we do, then we'll move them to our second group, which is the infection followed by vaccination group. So these are individuals who were initially infected, enrolled in the study, and then at some point during the study, typically between that day 90 and day 180 sample collection, were vaccinated, and we have received those post-vaccination samples. And finally, for our third group, we have our vaccination followed by infection group. So these are individuals who are vaccinated before enrollment in the study, then had a breakthrough infection and were subsequently enrolled. So all of the samples that we have both vaccination and infection influences. Overall, the cohorts are pretty similar with the age breakdown and with the sex breakdown. So to start off looking at the data, first we're just going to look at the longevity of the responses for our 3 different groups. We have our infection-only group, our infection followed by vaccination, and vaccination followed by infection. Along the x-axis, we have our days since enrollment in the study. For our infection-only group, we do have some samples that have that full year of collection, but for the other 2 groups, we only have out to day 180. And with that third group, the majority of those, we only have that day 1 and day 30 sample collection, so it's a little bit harder to accurately interpret the results. Along the y-axis, we are looking at Wuhan RPD titers and compared to the WHO standard, so therefore it's represented in binding antibody units per mil. And so what we see, starting off with the infection-only plasma, we see that peak titers that are reached around day 30, And then we see overall general waning of the antibody responses over time. I do have the groups broken down by high responders, fairly average responders, and then poor responders. And we see that with those that have very high response at that day 30, they typically do end up having a bit more rapid decline compared to those that are not as strong responders, resulting in that end time point having very similar titers overall. Next, looking at the infection followed by vaccination groups, we see once again that increase of titers around day 30, starting to see that waning of titers, and then we see that increase of titers again at day 180, with once again most of those individuals being vaccinated between that day 90 and day 180 time points, indicating that with vaccination we are able to see an increase in titers overall, and we are able to see an increase in the peak titers compared to infection alone, indicating that vaccination will hopefully be able to help us achieve higher titers compared to just infection. And once again, with the vaccination followed by infection, we don't have a ton of individuals in this group yet. We do have more samples coming, but we're able to see that starting off, they typically have a little bit higher titers compared to our other two groups. And then we do see a slight increase in titers at day 30, indicating that even with vaccination, if you have breakthrough infections, you're still able to see a bit of an increase in those titers. Though for our one individual that we do have out to day 180, we're still seeing that decrease in titers over time. In addition to longevity, we also want to look at the breadth of the antibody response. So we have heat maps showing the breakdown of our 3 different groups once again, and these are looking at the peak titers. So for our infection-only plasma and also our infection followed by vaccination plasma, these are typically derived from day 30 samples, and then from our infection followed by vaccination, this is typically day 90 or 180, whatever that time point is after the post-vaccination. Along the bottom of the graphs, we are looking at the different variants, Wuhan, Alpha, Beta, Gamma, Delta, Lambda, Mu, Omicron, as well as the nucleocapsid. Along the y-axis, we are looking at the individuals, and so whenever you look at the rows, you're looking at one individual's response to those different proteins, and whenever you're looking at the columns, you're looking at all the individual responses to that particular protein. Starting off with the infection-only plasma, we see that overall it's much darker, indicating that even at peak titers, individuals are producing much lower peak titers compared to those that have seen vaccination. Whenever you look at the Mu and Omicron columns specifically, you see that Mu typically has a little bit brighter colors than the others, whereas Omicron has darkers, which indicates that both shows the immunoevasiveness of Omicron, which has been reported, and also with Mu, once again, this is one of the variants that was circulating whenever a majority of individuals were enrolled, and so it seems to indicate, once again, that with infection, you have more of a specific response to the variant that you're exposed to, instead of producing a broad response. Whenever we look at our 2 vaccination groups, so either infection followed by vaccination or vaccination followed by infection, we're seeing brighter colors overall. And we're also seeing brighter colors across the rows, indicating that we're seeing a broader response whenever individuals have been vaccinated, indicating that you should have a broader protection with vaccination compared to infection. However, you'll notice that there are still some dark colors in the vaccination groups, indicating that there are some individuals, despite being both vaccinated and infected, are still really not producing great antibody titers overall. And this leaves us with the question of whether they're protected from potential reinfections. And we have been able to potentially capture some reinfections in our group, as determined by showing up PCR positive on day either 90 or 180 after having no detected viral burden on day 30. And so whenever I looked at these groups and looked at their systemic IgG titers, I expected to see that perhaps they would have lower titers overall, indicating that they may have had less protection. However, whenever we break apart the systemic IgG titers for those that are PCR positive versus PCR negative, and looking at those different groups of infected only and infected followed by vaccination, we really don't see much differences between these groups, indicating that there's really nothing about their systemic IgG titers that could indicate whether they are susceptible or potentially protected from future reinfections. Another concern with COVID is long COVID. We have almost 50%, so 41%, that have reported long COVID symptoms, in our cohort, with majority of them experiencing 2 or more symptoms, with some of those common symptoms being alopecia, fatigue, and headache. And once again, whenever we look at the systemic IgG titers, surprisingly or unsurprisingly with this, with long COVID, we once again don't really see any differences that could indicate whether an individual will potentially suffer from long COVID. So far, we've only been talking about systemic IgG titers. For a lot of the reports that we've been seeing in papers, they typically focus on systemic IgG, and so we're curious about what the mucosal antibody response would look like, particularly in the nasopharyngeal areas, because this is the site of infection. And so with our nasal swabs, postdoc in our lab, the brilliant Erica Rubidoux, was able to come up with this swabology workflow to allow us to be able to look at SARS-2 specific antibodies in the nasal swabs. So general overview of the workflow is first looking at the total protein concentration in the nasal swabs and then equalizing it to make sure that we're starting out with the same protein concentration for all the swabs, and then looking at the total IgA or IgG that's present in the swabs, and then finally looking at the antigen-specific IgA or IgG within the swabs. And in this case, we looked at anti-RBD and anti-N. And so first, we just wanted to see if we could detect any specific differences between our cohort with the mucosal samples. And we were able to see that there are some individuals that have a really robust mucosal response and others that really do not have a great mucosal response for IgA against anti-RBD, which is shown in the green, or anti-N, which is shown in the yellow. And interestingly, whenever we looked at the symptoms for these individuals, we saw that at acute infection, they're among the most symptomatic, with most of them having 4 or more, some of them up to reporting 11 symptoms, with those symptoms including loss of smell, loss of taste, runny nose, headache, malaise, and also conjunctivitis. And so then we're curious of what do our potential reinfected individuals also long COVID, what do their mucosal responses look like compared to the rest of the cohort? And so we only have a few of these samples. So far, we only ran about 40 of these samples. And so right now, it's pretty low numbers, but we did have some interesting things that pop up. Specifically, first looking at those that potentially have been reinfected, so those that are PCR positive on day 90 or 180, we ended up seeing that they had overall lower mucosal responses compared to those that are PCR negative at these time points. And we also saw similar trends with those that have long COVID. So whether this is just a phenomenon of being unlucky with the samples that we picked, or whether it actually is something that where mucosal responses could end up being indicative of protection is something that we are hoping to look at further. So to wrap things up, we have individuals that appear to be susceptible to reinfections and long COVID, regardless of the initial infection burden, systemic IgG titers, or vaccination status. But we do potentially have something popping up where mucosal IgA titers do appear to be lower than individuals who are not suffering from these conditions. And so we're hoping to explore further to see whether mucosal titers may be more indicative of protection or susceptibility compared to systemic titers. We also hope to compare this to our Memphis cohort to be able to see if this is translatable across different populations, and then also our Indy flu cohort, which is cohorts from Northern Columbia as well, looking at influenza to see if it's also translatable across different respiratory viruses. And so if anyone has any plasma and matched nasal swab samples that they'd like to share, we'd love to collaborate with you all. So thank you to the lab, particularly thank you to our collaborators, Juan Dib and the teams down in Columbia. This would not be possible at all without them. The CEIRR Southern Hemisphere Working Group, particularly Marlene and Anise, and also CEIRR's are on me to talk today. Thank you.
Do you have a few minutes for questions?
Hi, very nice talk. I just wanted to comment that Jennifer Gorman from University of Toronto sees a similar signal, right?
Mm-hmm.
It's not that strong, they don't have that many samples, but she sees that secretory IgA seems to be protective. Maybe reach out to her and maybe you can pull data to get a more significant signal out of it.
Yeah, that would absolutely be fantastic. Thank you.
So I have a question for you. I was interested in your infection-only group. Your young adult male group seemed to have a lot of people with very low titers. and the females did not. And so, did you have any thoughts about, is there anything else different? Do they have different risks of exposures? Or
Yeah, so actually, well, I guess I can't go back to that slide. So yeah, the infection-only group is actually very interesting because I'm not sure if you caught it, but it's for the males in the infection-only, for the young men, they're not producing great antibodies, and also for the older men. So there's 370 plus, so like 72, 75, 77, that are producing fantastic titers across the board with the infection only. And so we haven't started exploring further, but we do want to look to see what is causing, because it's almost opposite of what we would have expected to see. So that's something that has stood out to us, and we hope to explore further.
just a quick follow-up before we go to Tomer. Have you seen any similar patterns in the IgA
For the systemic IgA, or
for the mucosal IgA
For-
men versus women?
so we haven't looked at too much because we only have ran, like I said, a few samples. We finally have kits that are being shipped, and so once we have more, then we're going to end up running the rest of it. So we want to run the rest of the whole cohort for all time points to be able to explore further. But right now, the numbers are so low to where there's not anything that we could actually pick up any major differences. Like I said, I wasn't expecting to see anything with the reinfection and long COVID groups, but then they all ended up having pretty crappy titers overall. And so then something that we wanted to explore further.
I think that was really interesting. I would urge you to also look at IgA and serum.
I'm going back and putting plates today for that. So. Thank you very much.
All right, I think that concludes session 5. I believe we have some moderators for session 6 on their way. One more thank you to our speakers. Another round of applause. Great, so welcome to session 6, the last sort of data presentation section of the meeting today after a long morning. So I'm Sophie Valkenberg from the University of Hong Kong / University of Melbourne, and this is Rob Middleman from St. Jude's Research Hospital. And we've got our first talk today from Annette Fox, also from the University of Melbourne, talking about influenza vaccine responses among young children first exposed to influenza antigens by infection versus vaccination. Thanks, Annette.
Up at the moment. Shall I shut down this last one? Yeah. Okay. Yeah. Can people see my slides now? Yep. Okay, thanks everyone. I'm from the WHO Influenza Collaborating Centre in Melbourne and from the University of Melbourne that are at the Peter Doherty Institute. And I'm very grateful to the organisers for giving us the opportunity to present our talk or present our study on how first exposure to influenza via infection versus vaccination affects subsequent antibody responses to vaccination. So the background of this study revolves largely around avian influenza H5N1 and H7N9, which have infected around over 800 and 1,500 people globally, with case fatality rates of 56% and 39%. So, and very recently another case in the US. So the median age of H5N1 cases has been around 18 years, and only 10% have been aged greater than 40 years, whereas the median age of H7N9 cases has been 61 years, with 42% aged greater than 65 years. And as you all know, the influenza A subtypes have been classified into two phylogenetic groups based on the similarity of the HA stem, with group 1 including H1N1 and H5N1, and group 2 including H3N2 and H7N9. So ecological studies by Katie Gostick, who's just there, suggest that early life exposure to H1N1 versus H3N2 viruses may imprint protection against group 1 versus group 2 viruses respectively, and in part account for the differences in age groups affected by H5N1 and H7N9 in that older people would have been first exposed to H1N1, whereas younger people to H3N2. So the other part of the background to this study is that it's becoming more common for countries to recommend and fund annual influenza vaccination for children from the age of 6 months to 5 years. Therefore, it may become more common that people are first exposed through vaccination to a mixture of influenza subtypes. And it's not really known how first exposure to influenza antigens via infection versus vaccination affects subsequent responses to vaccination. Bearing that in mind, our aims of this pilot study were to compare immune responses to influenza vaccination in young children who had prior exposure through infection only, vaccination only, or neither. And we had some other aims that we don't really address so much today, but they were to characterise the immune response to influenza A virus infection in infants and young children, and to investigate really if influenza H1N1 versus H3N2 imprint B cell and antibody responses towards group 1 and 2 viruses respectively, and if this is associated with the B cell receptor heavy change in gene usage such as BH169. So the reason we can't talk much on that is because of the lack of cases with the study going through COVID and shutdown and lockdowns. So our study plan was to recruit 10 children aged 6 to 60 months in these different prior exposure groups, and to follow them across 2 years of vaccination by collecting blood on days 0, 7 and 28 of vaccination, and by examining HI antibodies in plasma and HA reactive B cells in the PBMCs. So we ended up recruiting 9 children. Just hit a pointer. We ended up recruiting 9 children with prior infection, 8 with prior vaccination, and 2 with no prior exposure. We recruited the prior infection group across 2019 through to 2021, whereas the prior vaccination group were almost all but one recruited in 2020. The ages of these children, or the birth years of these children, ranged from 2015 through to 2020. And some of the children in the prior infection group, a few of them received 2 doses, as well as both children in the naive group received 2 doses of vaccine. 3 of the children in the prior infection group were followed over the 2 years as we wanted, and 5 in the prior vaccination group, none in the naive group. So it's very, very hard to retain children into this type of study. So in terms of the demographics, the children who had prior infection were generally younger than those with prior vaccination, and the naive ones were very young. In terms of the prior subtypes that cause infection, I should have pointed out we only were looking at children who had prior influenza A confirmed infection. So subtypes were H1N1 for 3, H3 for 1, and they weren't subtyped for the rest. And there wasn't really an age difference there by those subtypes. The interval between vaccination and sample collection, it was longer than the 7 days and 28 days post-vax. So some of the children in year 1 in the prior infection cohort and in the naive children, otherwise all the samples were collected in a very timely way. The sera were tested against 7 H1N viruses, 12 H3N2 viruses, and 4 B viruses that represented strains that were circulating during the children's lifetime, as well as the vaccine strains that they were getting, or the prior vaccine background viruses. So I'm just going to start with all the H3N2 serology and then move on to H1N1 serology, and just first up to show really raw data against one virus, and this is against the 2020 vaccine strain. So most of the children, or all of the children in the prior infection group and the prior vaccination group showed a titer rise post-vaccination, but the 2 naive children did not, or very little, and they had low titers. And when we look at summary statistics, though, we can see that while children in the prior infection group had similar pre-vaccination titers to those in the prior infection group, other way around, infection and vaccination groups, the post-vaccination titers were substantially higher in the prior infection group, and they were very low in the naive group. And this translates to substantially higher titer rises here post at the 2 day 7 and 28 time points among the prior infection group compared to the prior vaccination group, with every child having a greater than 4-fold rise in the prior infection group and half of the children in the prior vaccination group having less than 4-fold rises. So now moving on to look across all those viruses that we tested, pre-vaccination, there really isn't much difference between the prior infection group in pink and the prior vaccination group in green in their pre-vaccination titers across those strains. But post-vaccination, there is a difference. Like the titers are higher against most of the strains in the prior infection group than the prior vaccination group. And this is particularly notable for some of the cell-grown viruses having quite the largest differences. And this also again translates to substantially higher titer rises across a lot of strains in the children who had prior infection. This is all year 1 data. So, and again, those cell-grown strains having the biggest differences. And so to try and then sort of amalgamate it and look at across years, we then look at titers averaged across all cell strains or all egg strains. And the biggest take-home message here is that in year 2, there is still some difference between the children who had prior infection, shown in pink, compared to those with prior vaccination in terms of having a bit of a higher titer. Now, unfortunately, there's only 3 children in the prior infection and 5 in the prior vaccination group at this stage, so nothing is significant. And again, with titer rises, that's the same thing. In year 2, there is some difference, but it's not significant anymore. So summing up for the H3N2, we found that mean antibody titers were similar before vaccination, but substantially higher post-vaccination for previously infected compared to previously vaccinated children. Titer fold rise is more than 5 times higher for previously infected compared to previously vaccinated children, even if prior infection was with a H1N1 strain. So that's quite interesting. So, and that was sort of suggests that the titer rise difference could potentially be a consequence of greater priming of T helper cells by infection than by inactivated vaccine, and that's something that the Rochester group, Jennifer Nyack's group, has shown before in children. Just interestingly, also in adults, we haven't seen that prior H1N1 infection has any influence on titers against H3N2 viruses or vaccine-induced titers against H3N2. Naive children had really low titers and negligible titer rise despite receiving 2 doses. Results suggest that vaccination is relatively poor for priming vaccine responses against H3N2 viruses, and in turn, that vaccination is relatively poor at generating memory B or T cells that mediate boosting. So moving on now to H1N1 serology in the same sort of format. So again, most of the children showed some sort of titer rise, and particularly in the prior vaccination group this time, unlike H3N2. A couple of the children in the prior infection group did not have really much response at all. And the other notable thing is that they, despite having prior infection, they all have really low, well, all but one have low pre-vaccination titers against H1N1. And again, naive children don't really respond very well. And so in terms of the subtype that was causing prior infection, was it H1N1? It's actually, and what's really surprising is that those children in red who had prior H1N1, as opposed to H3N2 or unknown, had really the poorest titer rises against H1N1 after vaccination. And again, going to summary statistics, we can see that there is a difference in pre-vaccination titers, with the prior vaccinated group having higher pre-vaccination titers against H1N1. But after vaccination, they sort of catch up. The group with prior infection catch up to the prior vaccination group. And that's sort of the same thing again with titer rise. Because they're catching up, you're seeing this really nice titer boost in both the prior infection and vaccination groups and not the naive children. And looking across strains, like for H3N2, you can see that titers are higher across a range of strains for the previously vaccinated children in green compared to the infected children in pink. And it's notable that these differences are really for the egg-grown strains in this case. And post-vaccination, the previously infected children had caught up a bit with the previously vaccinated children. And this is just looking at now moving on to looking at this next year's data and looking at average titers across all cell or all egg viruses. You can see that really the children by year 2, the previously infected and vaccinated children don't look different, so they've really caught up. And same, moreover, actually by year 2, the previously vaccinated children with the pink colours have now had even higher titer rise than the previously infected have higher titer rise than the previously vaccinated children. So we're seeing that prior infection is better at priming vaccine antibody responses against H3N2, but not H1N1. And this is not the slide. Okay. So this side-by-side comparison further shows that if you look at the titers against H3 versus H1N1, and also looking at averaged across all viruses or just at some individual viruses, you can see that the group that had infection have the higher titers, have much higher titers against H3N2 than H1N1, whereas the group with prior vaccination, they have very similar H3N2 and H1N1 titers, and also the geometric mean fold rises are greater in the group with prior infection against H3N2 than H1N1. And the other interesting or intriguing thing about this is that we see this data is that we see these children who have really quite a polarised response with a lot of antibodies against H3 and hardly any against H1N1, or vice versa in the vaccinated group, in the prior vaccination group. And that's sort of just highlighted a bit more by pulling those children out, these sort of really polarised differences in how they respond to H3 or H1. So to sum up the serology for both H1 and H3N2, so this was a pilot study and we didn't have sufficient children really to look at how H1N1 versus H3N2 might imprint infection. It was conducted during COVID, so our lockdowns meant that we couldn't really investigate current infections. Our low sample size also limits interpretation. However, we do see that previously infected children had negligible antibody against H1N1, even if the prior infection was with a H1N1 virus. So this could, like with these low sample numbers, it could be a chance finding, but we looked at the WHO serum panel data over a number of years, and this is consistent with that, where children under 36 months were having lower titers against H1N1 than H3N2 prior to vaccination. So it seems to be a bit of a phenomenon that children aren't having a priming through infection for having H1 antibody levels. I mean, it's not just immature immunity because they have H3N2 antibody responses. So although primary infection didn't seem to induce or sustain H1 reactive antibodies, it did seem to prime the antibody response to vaccination, since titer rise against H1N1 was similar among children with prior infection versus vaccination, and substantially greater than in previously naive children. So this sort of paradoxical effect of infection on H1N1 is similar to findings for LAIV. So it sort of raises the question of whether infection is inducing H1N1 HA reactive memory B cells without yielding long-lived antibody secreting cells. Or, there's something about having exposure to all 8 proteins when you have an infection as opposed to just having mainly HA during vaccination. So the results suggest that infection is substantially better than vaccination for priming vaccine-induced antibody responses against H3N2, whereas vaccination appears to be as good as infection for for priming vaccine responses against H1N1. So where do we go from here? Our results for H3N2 are quite analogous to our studies in adults, where vaccine-induced antibody responses are enhanced by prior H3N2 infection, but not by prior vaccination. So we sort of theorise, along with being stimulated by Scott Hensley and Sarah Kobe's work, that vaccination provides limited priming of naive H3N2 HA-reactive B cells and largely recalls memory, whereas infection robustly primes naive H3 HA-reactive B cells, generating memory against an array of epitopes. And we want to explore this further, this epitope range of the H3N2 reactive B cells and antibodies in these children by analysing the B cell receptors used by H3 HA vaccine strain-specific and cross-reactive B cells, and by measuring HI antibodies against viruses bearing wild-type and mutant HA, and HA mutated at individual antigenic sites. So I'm just trying to squeeze this in. So this is just an example. These are examples of what we're doing there. So we're looking at the children's responses to reverse genetics viruses containing HA from an egg-grown Hong Kong 4801 2014 H3 virus. So just the HA has gone in there, or we've mutated that HA at site B by switching in H10 amino acids. This is a process that was described by Palazzi's group. And so those mutate positions in critical antigenic sites. And we've also looked at reverse genetics viruses containing cell-grown Hong Kong virus HA. So this is sort of data we get, and the way we analyse this data is we see what is the fold rise pre to post vaccination against the wild type versus the mutant virus. So in this case, it's not that different. And then what is the fold reduction in titer against the mutant virus compared to the wild type? So here you've got an 8-fold reduction or a 4-fold reduction. So we're sort of putting out these little heat maps, or what are the fold changes for the different viruses there. So in that case, you couldn't see it very much, but when you went on to 2021 CEIRR, or the 2021 vaccine response in this same child, we start to see quite bigger differences in fold rises. So there's 8-fold rises against the wild type, but only 2-fold rises against the mutant viruses. And in addition, you started to see much bigger fold reductions in titers against the mutant virus compared to the wild type virus, especially at the post-vaccine time points. So that would indicate that you're getting increasing amount of antibody being focused on site B. Okay, I better finish up. So we've sort of gone through this. We see some children with evidence of focusing and some without. And I'll go through that quickly. And so putting it all together, what we sort of think we're seeing so far is that in the children with prior vaccination, there's very few who already have the antibodies responses focused on site B prior to vaccination. But then a lot of them, they start to get increasingly focused as they have more vaccination. In the children with prior vaccination, a lot of them don't really have that much greater response anyway. But in those that do have a lot of antibody, it's already quite focused on site B, and it can increase after an additional vaccination. So the other things we want to do is to sort of look more into this paradoxical effect of H1, of prior infection on H1 titers. And I guess the final take-home message is that, these relatively poor responses, especially to H3N2 and the poor responses in naive children, indicate that we need to look for vaccines or use vaccines that are better able to prime immune responses across subtypes in young children. And there's already a number of studies on adjuvanted vaccines that would say that they can do that better than just inactivated vaccine. And so I'd just like to finish by acknowledging everyone who's helped with this work, in particular Kuan-Fu is the PI for this study and very critical for getting funding for us all. And the people in my lab who've done a lot of the serology, Louise, Steph and Jess, Yu-Yang Seng or Ryan Seng, he's created the reverse genetics viruses. Sheena and her team for helping us with data analysis and always giving me code that I can use. And very importantly, their children and their carers, the hospitals, the Royal Children's Hospital and Westmead Children's Hospital, where children were recruited, and the PIs there, Nigel Crawford, Phil Britton, and the coordinators, Annette Alafaci and Rebecca Burrell, and thanks to the Rochester CEIRR program for funding this study through an auction. Thanks very much.
Thanks, Annette. Fantastic work. I think we can't ignore Derek Smith for one very quick question.
Thank you. Annette, was this an egg-based vaccine?
Yes, it's an inactivated egg-based vaccine.
So that H1N, that H1H3 difference, perhaps I missed, but it could also potentially be caused by the fact that the H3 in the vaccine had lost glycosylation in the egg vaccine and the H1 not. So might that explain the difference?
I'm not sure that it would. I mean, they did really respond really well to the vaccine. To the H3 and 2 antibody responses were very, very good. So, and even if they were infected first, which would mean they'd see more of a cell-like virus, and the vaccine's egg-like. So, they would have had, maybe they would, I don't know, I mean, they wouldn't have had antibodies directed towards that glycosylation episode after their infection if it was with H3 and 2, but the infections weren't necessarily even with H3 and 2. So,
Okay,
I don't think it would, no.
Thanks.
Maybe we need to talk more about that.
Thanks, Annette, and welcome, Katie Gostick, for differences in the cross-reactivity of ferret and human anti-CEIRR. A great segue for the imprinting routine.
Okay, very good. So, I'm Katie Gostick. I'm a research scientist in Sarah Covey's lab at the University of Chicago. Really want to thank the organizers for the opportunity to come tell you today about differences in the cross-reactivity of ferret and human anti-CEIRR. Okay, so for pathogens like flu and SARS-CoV-2 that can evolve to escape host immunity, cross-reactivity between established strains and newly establishing variants is this really powerful force that shapes things like population immunity, strain fitness, and vaccine effectiveness. And for flu, the main way that we measure cross-reactivity between strains relies on measurements from primarily infected lab ferrets. So the basic idea is you take a ferret, you infect it for the first time with a known strain, and it raises a primary antibody response that's specific to that strain. Then we can take these antibodies and test them using HI or neutralization titer measurements to figure out how much worse they bind a collection of heterologous strains as opposed to the homologous strain. And so these are the main data that are used to estimate things like the antigenic distances that we see in antigenic maps. They inform evolutionary forecasts in next strain, and they're also one of the main pieces of evidence that WHO uses to assess whether newly emerging or circulating strains continue to cross-react with the existing vaccine strain. Okay, so essentially what these ferret measurements do is they tell us something about a virus's antigenic phenotype and the binding properties or differences in antibody binding between two viruses. But I think often in practice, we use these antigenic distance estimates or these ferret titer data as a proxy for how well a new variant will escape host immunity. And in this context, this antigenic phenotype is really only half of the puzzle, right? Because we know that the human population is diverse in age and sex, infection history, genetics, and all kinds of other immunological factors that could cause hosts to respond differently to the same antigenic change. And so really the question that we're trying to get at is how these ferret titer data fit into this picture once we start accounting for host heterogeneity. And I want to emphasize that this is not just a hypothetical problem. For the sake of time, I'm not going to go into too much detail describing these examples, but I do want to highlight that there have been some really poignant recent examples in which a new antigenic variant of flu comes onto the scene, and we test its antigenic properties using ferret anti-CEIRR, and we don't really see that it's antigenically distinct. So the ferret CEIRR continue to cross-react well. But when these viruses start causing epidemics, sometimes we see that certain subsets of the host population have lost immunity. And this can happen either if individuals in the population are targeting different epitopes than the ferrets, or if immunologically experienced hosts are recalling responses from memory that can still bind those viruses but don't neutralize them. Because the ferret antibody responses are primary responses, this isn't sort of a recall effect that we would expect to see in the ferret sera. Okay, so this leaves us in a situation where we have to acknowledge that new oncogenic variants are not necessarily always escaping immunity equally across the host population. We know that our cross-reactivity measurements aren't designed to detect this kind of immune escape, and sometimes this can obscure signals of epidemic risk or prevent us from noticing that a vaccine update might be helpful. And so I think the big picture questions that we think about a lot are whether this kind of cohort-specific immune escape is common, or whether the examples I just highlighted are just that, sort of unusual examples. I think it's also really important for us to figure out what factors define a cohort. We talk a lot about immune history or birth year, but there are all kinds of other individual differences that could cause hosts to respond differently to an antigenic change. And ultimately, what we want to figure out is how much of a problem is it that these existing cross-reactivity measures can't really capture diversity among hosts. Okay, so these are very broad questions, and I'm not going to fully answer them in this talk. Excuse me. So mostly what I'm going to do here today is I'm going to talk about how we're approaching these problems. I'm going to show some analyses that we consider preliminary, and then I'll talk in detail about the data that we think we would need to take this kind of analysis to the next level and get more comprehensive and definitive results. So what we're going to try to look at today are some very basic empirical questions. First, how accurately do these ferret-based measurements capture the average human being's cross-reactivity to a new strain? Second, how variable is the level of cross-reactivity that we see in new strains among humans? And third, I'm not really going to talk about this because we don't have rich enough covariate data, but ultimately what we want to try to do here is identify individual level covariates that can help us predict who in the host population is most likely to show lower cross-reactivity than a ferret. And the way that we're doing this, the general approach is that we're taking anti-sera from previously published data sets, and we can use those to estimate what we call individual distances from human antibody responses. And so the individual distance is defined as the difference between an individual's homologous log2 titer and their heterologous log2 titer after infection or vaccination with a known strain. And so this definition is sort of drawn from established theory and methods used to construct antigenic maps. And so we can estimate these individual distances from human anti-sera, and then we can compare them to the more canonical ferret-based map distances that had been estimated previously. And so we're taking human samples from 3 sources, and these are all from previously published studies, mostly by Judy Fonville. And so the first source of data that we're looking at focuses on human samples after primary infection. And so these are taken from a study published in 2016 that was specifically trying to compare antigenic maps that would be estimated using anti-sera after primary infection in ferrets or in children. And so I do want to say that the sort of one main weakness of these data is that these samples are taken from a blood bank from a children's hospital in the Netherlands. And so we don't really know, we don't have confirmed primary infections in these individuals, but the samples were deliberately filtered in a way that makes it likely that these children had a recent primary infection based on the age that they were when the sample was taken, based on sampling during a period of H3N2 circulation, and based on pre-screening for seropositivity to H3N2. And then we're also looking at anti-sera sampled from 12 individuals in the Hanan cohort that had a PCR-confirmed infection in the past year, and then 234 individuals that were part of an independent vaccination study. Okay, and so I've already mentioned that there are some limitations of these data, and so I'll talk in greater detail at the end about what we think we would need to do this even better. Okay, so diving into the results, one of the first things that we see is that we can use these ferret-based distance estimates as a linear predictor of the individual distances which are being shown on the y-axis. And so the regression line basically shows the relationship between the ferret-based distances and the human distances. But you can see in this figure that no matter what serum type we're looking at, which are shown in the rows, or no matter whether we're fitting to antigenic distances to strains that circulate in the future after the homologous strain, or in the past before the homologous strain, across the board, these fits explain very little of the variance among the individual distances. So our R-squared values are always pretty close to 0. And I'm also showing the estimated coefficients here, which are also sort of close to 0. And so there are a couple things that I want to pull out of this figure. So first of all, this is not surprising, but in secondary, but not so much primary human anti-CEIRR, we also observe a lot of negative distances. And this is especially true in the vaccination study and when we look at individual distances to past strains. And so basically this means that these folks have higher titers to strains that circulated in the past than to the strains that they were most recently exposed. And given everything that we know about recall of immune memory and original antigenic sin, I don't think this is super surprising. But operationally, it's important to understand that individual distances to past strains should be interpreted differently than individual distances to future strains. And operationally, this also means that if we want to incorporate more secondary anti-CEIRR into these kinds of comparisons, we really need to be using strain panels that prioritize forward temporal breadth, that prioritize strains that circulate after the time of most recent exposure. And so that means that we need to be going back into the freezer and pulling out samples from individuals that were infected or vaccinated with strains that circulated deep in the past. Well, maybe not that deep, 10 or 15 years, we think should do it. Okay. And so the other thing that I want to highlight is this issue of individual variability. So we already talked about how these ferret-based distances shown by the regression line aren't really capturing individual variability in the population. And so I've taken the confidence interval in this figure and I've replaced it with this ribbon that highlights fourfold differences from the central ferret prediction. So we usually think about fourfold differences in titer as antigenically or immunologically meaningful. And we can see that for the vaccinated cohorts and for the primarily infected children, there are definitely some individuals that fall out of this fourfold ribbon, which either means if they're below the ribbon, that they show much more cross-reactivity than we'd predict based on the ferret distances. Or, I think more importantly, there are a large fraction of data points that fall above the ribbon, meaning that these are individuals that are showing levels of cross-reactivity that are much weaker, more than fourfold weaker than we would predict based on the ferret data. And I'm not showing this in the slide, but I've also looked through this, and being one of these data points is definitely individual associated. So there are a lot of individuals whose individual distances to different strains on the x-axis all fall within the ribbon. So these folks are pretty ferret-like. But then if an individual has one data point that falls up in the low cross-reactivity range, they're more likely to have more than one data point up there. And so we need richer covariate data and bigger sample sizes so that we can try to figure out not just if this is random noise or random variation among individuals, but variation that we can explain. And it's possible that we might be able to identify covariates that allow us to better predict who in the population will be vulnerable to new antigenic substitutions or to vaccine mismatch. Okay, so to do this, I said we do need human anti-CEIRR samples with rich covariate data. Ideally, we'd have a substantial number of samples per season, and we'd be able to look across multiple seasons, right? Because we know that mechanistically, the factors that drive antigenic escape can really differ from year to year. And we've already talked about how to do this, we really need to be using strain panels that prioritize forward temporal breadth. Okay, and so I'm bringing this figure back because the second thing that I want to highlight here that we've seen consistently and think is worth noting is that the ferret-based distances systematically overestimate the distances we see in humans. So this is the same figure we showed in the previous section, but now I've added this dashed one-to-one line And so if the raw unadjusted ferret distances were accurate and unbiased predictors of human cross-reactivity, then we would expect the individual distance data points to cluster around this one-to-one line. But instead, what we're having to do is basically use a linear regression that really lowers the slope of the relationship between ferret and humans. So basically, on average, we're seeing stronger cross-reactivity in human anti-sera from any of these three groups than we see in ferrets. And this isn't an independent validation of this result because we did reanalyze data from this study, but I also want to point out that we're definitely not the first to remark on this pattern. So these are figures from that Fonville et al. Study published back in 2016 that compared antigenic maps constructed using ferret and human anti-sera after primary infection. And so this study argued that, overall, the general layout of the map in terms of how the different colored strains cluster is similar when you construct the map using sera from humans and ferrets. But the inferred strain locations, and more importantly, the distances between them differ. So the ferret map has a greater spread than the human-based map, indicating that on average, the ferret distances are greater. Okay, and so it's possible that this is a biological species difference between ferrets and humans, but we think it's really important to follow up on this pattern and try to figure out whether we can explain these differences between ferret and human cross-reactivity as sort of a confounding effect of how the ferret data are collected. So most of the ferret samples are collected 3 or 4 weeks after exposure, which is right near the peak of the antibody response. It's also a time when affinity maturation might still be in progress, and so it's possible that if we waited longer, that the ferret anti-sera would show greater cross-reactivity due to affinity maturation and also lower distances as those homologous titers, which is sort of the thing that we're subtracting from in the individual distance calculation, as those homologous titers start to get lower. And so we'd really like to be able to re-estimate distances using ferret anti-sera collected at different time points post-infection, maybe going all the way out to a year if anybody has those samples. And then I think it would also be useful to think about route of exposure, right? It would be cool to compare distances estimated in ferrets that are experimentally inoculated with a high dose versus more naturally infected after contact with a donor. And again, if we want to do this, we would need to be able to test against a strain panel with forward temporal breadth, meaning that we would probably have to do it using ferrets that had been exposed in the past. Okay, so wrapping this up, we've talked about how our existing methods to estimate cross-reactivity between flu strains are really designed to capture the antigenic differences between strains, but not to capture host heterogeneity in the population. And sometimes this can be a problem, but it's not clear how systematic the problem is. And so we've seen that when we look at individual variability in comparison to these ferret distances, there's a lot of variability among individuals in terms of the level of cross-reactivity we observe to new strains, and we need richer covariate data so that we can try to figure out whether this individual variation is predictable or whether it's just random noise. I don't think it's just random noise. And then finally, we talked about how ferret-based distances seem to systematically overestimate human distances or underestimate human cross-reactivity, and I think that it would be a good idea to dig into this and try to figure out if it's an issue of sample timing or dose, or whether it's more of a biological difference between these species. Okay, so with that, I want to say a huge thank you to everyone in the COBE lab who's given really great feedback on this work, and especially Sarah. And I want to acknowledge funding from Penn CEIRR, and hopefully we have time for questions.
Thanks, Caitlin. Great job. It's right up to Derek.
Thank you. Caitlin, it's really great that you're doing this. Yeah, I completely agree with what you're talking about. Sam Wilkes in Cambridge has been looking at similar things. I think he'd be really interested to talk to you. Sam worked very closely with Judy as well on a number of things. The other thing is what you talk about affinity maturation. Yeah, indeed, may very well be. And for us, one of the things that's been fabulous about SARS-2 is that we're all kids to SARS-2.
Yeah.
Most of us are kids to SARS-2. And there's a hell of a lot of data. And you can also get more blood as well because you're taking it from adults. So I think this is a really great place to look as well. And it's absolutely clear that 3x vaccinated individuals, for example, they have much different slopes than 2x vaccinated individuals. And so it's really also a very rich data set for looking at this stuff.
Yeah, that's really interesting. Okay, maybe I'll talk to you later.
Yeah, hi Katie, that was fantastic. So first we have ferret samples that go out of here. So, and I know you know that, so happy to chat about things that we can do to help with that and sort of hole you have. But I was actually thinking even more about other ferret samples. If you could potentially speculate on how many human infections might be driving some of the negative correlation, obviously, that you see with the adults that have multiple infections. You know, we have so many in our lifetime. Whether you think it's the primary one that's really driving it, or whether it's like 3 or 4 different infections that then will change the relationship, because we could use, you know, the ferrets that have been sequentially infected in my lab or that get natural infection with prior immunity to sort of help assess whether that, you know, 1 or 2 infections can really be driving it. Do you get what I'm trying to say?
I think maybe, so you're talking about where I was showing those negative distances.
Yeah.
I actually think that if we redo this with ferrets, like primary infections might be the place to start. I think that those negative distances, I mean, it could literally be as simple as the fact that this person has been exposed to multiple strains in their lifetime and we're tested, like, it could just be a homologous response from the past. You know what I mean?
Right.
Yeah.
Okay.
That's good. Very solid talk. Sorry, Toma, but just to keep for the next talk, can we still have our tea break? Great. And I'm going to introduce now Robert Middleman from the St. Jude's, who's going to be talking about defining cellular correlates of protection to influenza across human cohorts. Thank you.
All right, thanks Sophie for that introduction. So I didn't have to introduce myself. I'm Rob Mettleman. I am a postdoc in the lab of Paul Thomas here at St. Jude. And I'm really excited this morning to tell you about some of the work I've been doing on several of the cohorts that we have access to through the CEIRRs and CEIRR networks, focusing specifically on a cohort that was introduced to yesterday to one group to cohort. Okay. So this doesn't need too much introduction, so I'll go through it quickly, but really what we're interested in is understanding the determinants that allow, just a second. Okay, I'll just use the enter then. Okay, so really what are the determinants, either the cellular, innate, T-cell mediated, or humoral responses that really dictate whether or not an individual develops no or asymptomatic infection when exposed to flu, or develops a symptomatic infection that could progress to severe, more severe infection. And so typically when we discuss correlates of protection, we're referring to antibody-mediated protection. This is kind of the historic measure of whether or not a response to a vaccine or post-infection is really going to predicate protection or not. And these are due to antibodies that are generated against the hemagglutinin head. There's been a lot of really good data recently about antibodies targeting the hemagglutinin stalk, as well as the neuraminidase. In our lab, we're actually interested in the cell-mediated components of this. So aside from the antibody responses and the pre-existing humoral immunity, what cell components are also contributing to this protection? And are they, in fact, independent or synergistic with the antibody responses? There was a study done during the 2009 pandemic, which gave us really good insights into this. And this was done in individuals who had not been exposed previously to the novel H1N1 strain at that time, but had pre-existing cross-reactive CD8 T-cell immunity. And that cross-reactive T-cell immunity, independent in the absence of antibody protection actually afforded them protection. Our group and others have also looked at cellular factors that are involved in reducing severity, so MDCs and IAV-specific T cells, as well as those factors, predominantly inflammatory cell and inflammatory cytokine mediators that are involved in increased severity. So in our study, and what I'm going to discuss today is really to determine these baseline cellular correlates of protection and severity across a vaccinated and unvaccinated population, making these statistical adjustments for both demographic and antibody titer information. And the key point here is that we're actually using baseline cell collections. So these are pre-season for the unvaccinated and 14 days post-vaccination for a vaccination group. So the quick experimental questions, as I mentioned, looking at the unvaccinated and vaccinated cellular correlates of protection, comparing how these correlates of protection may differ across vaccination status. And then in the vaccinated individuals who do progress to symptomatic infection, can we define predeterminate cellular factors that are correlated with that vaccine failure? And then finally, how do we use these data? Well, we want to understand whether or not we can predict or identify individuals that are at increased risk of symptomatic flu. And here's our cohort. I won't spend too much time on it because we got a lovely introduction to it yesterday. These are the Shivers 2 cohort, also known as Well Kiwis. It's housed in Wellington, New Zealand. And the sample study set that we're using is a selected 206 participants. I'm showing their age, biological sex, and vaccine status that they've selected, as well as the infections that we observed amongst those 206 participants. The majority in that year were flu A, H3N2 specifically. We also had 2 influenza B infections, and then we have 14 cryptic infections. And these are infections that are defined not by PCR positivity, but seroconversion pre and post season. So asymptomatic, those will become important later. Okay, so a little outline of what the analysis pipeline for this study looks like. We have a whole wealth of data collected by the Shivers team. We have demographics listed here, vaccine histories, infection status, PCR status, and strain. We have HAI and NAI titers for serology, and our lab was really involved in generating the cell frequency data by high-dimension flow cytometry. The analysis comprises univariate, predictive, and multivariate statistical modelings. The univariate, I'll just share some quick categorical and continuous variable analyses and go into a logistic regression. We also use a little bit of predictive modeling to understand how best our covariates are able to categorize an individual pre-season as either symptomatic or asymptomatic flu infection. And then we bring all of these demographic, vaccine and serology and cellular components together into a multivariate statistical model to predict risk. So the first analysis we looked at was correlations between demographics and serology. I think this is really interesting. If you look at the yellow bars, those are individuals that have a unprotected antibody response pre-season. So in the unvaccinated, we can see that the majority of individuals are unprotected based on their HAI titers. On the bottom left panel, you can see that once those individuals receive the vaccine, about 50% of them generate protective responses across the board, but that still leaves about 50% of the participants that are not responding to that vaccine from HAI titer standpoint. Across the board for unvaccinated and vaccinated, interestingly, we do see the majority of individuals that have protective NAI titers. So if we look at correlations between participant age on the top and BMI or body mass index on the bottom, we don't see much correlation at all with either the HAI or NAI titers. This is showing the NAI titers. Based on the graphic on the left, we know that the majority of the responses are protective, so the majority of the variation occurs above that dotted line, which is an AUC of 40 or protection, but we don't really see much correlation. I think there's only one significant correlation with NAI titers to H1 in the female participants here. So we next wanted to look at the cellular components, and this was kind of the big contribution that our lab made to this study. And so what we did was we took those PBMC samples, we stemmed them with peptide or live virus with common flu epitopes, And then what we were able to do was use high dimension flow cytometry using a spectral analyzer to analyze cells across the lymphoid and myeloid cell populations. And this allowed us to determine cell frequency, do surface immunophenotyping. Using those STIM conditions, we could pull out antigen flu-specific responses, and using the ICS, we know what those responses look like at a cytokine level. And to walk you through this quickly, this is a few representative populations. We had over 89 populations that we analyzed, and I didn't want you to all squint. So we're just going to look at several populations that are significantly increased, in these three cases here, significantly increased in known or asymptomatic infection, shown on the left here. And then one particular population, this activated NK cell population, which will come up a few times in the talk, that is increased in symptomatic flu cases across vaccinated and unvaccinated individuals. And to take all of these together, we did a logistic regression analysis, just looking at each individual population. This is a univariate analysis, so each individual is analyzed separately. And so the populations that have values that are negative in this log transformed odds ratio actually reduce risk of symptomatic infection. So some of these populations are dual cytokine producing CD4s, dual cytokines producing CD8s, as well as some other lymphoid populations. Populations that are on the right or positive values are significantly increased or increased risk of symptomatic flu infection. And those include, the scale got kind of messed up with these CD4s. They're so strong, but cells within the innate populations are enhancing risk at a univariate level, and then some lymphoids, including gamma delta T cells, are also increasing risk. So one of the questions we always kind of get with these types of analysis is, if you look at protection amongst asymptomatic and no infection, the no infection is kind of a problematic grouping because you don't actually know if they've been exposed to the virus or not. So what we wanted to do is further subset the data and just look at those that we know have been exposed to the virus. So those are the asymptomatic infections, their exposure is confirmed by serology, and the symptomatic, which we know by PCR were positive. If we do a logistic regression with non-cellular factors, we can see that the only factor that's really skewing towards protection are pre-existing levels of NAI titers against H1N1. These other factors do not reach any level of significance. At the cellular level, we do see two populations, again, activated NKs and NKs producing interferon gamma that are significantly increased in their cell frequencies amongst symptomatic participants. In asymptomatic participants, we did actually have quite a few that were associated or increased with asymptomatic infection. Several of them are shown here, but the list is quite long. So what this says is that not only are there differences in responses, but the baseline cellular profiles may actually preclude either symptomatic or asymptomatic infection over the course of the season. Okay. We know the immune system is just that, it's a system. And so we wanted to understand how groupings of immune cells could cluster together to represent co-regulated immune cell modules. So groupings of cells that are responding in tandem have the potential to respond in tandem. And so we did this across the myeloid compartment using a Pearson correlation. So along the central diagonal, you can see clusters of highly purple or highly positively correlated cell populations. We reduced our population number down to 9 individual clusters. And then across the lymphoid population, you can see a number of clusters. I think there's 13. So what this tells us is that these baseline immune responses are actually very highly correlated with one another. And those modular responses will be important for understanding how groupings of cells are responding to influenza vaccination or infection. And we did a similar type of analysis where we took the average frequencies amongst these clusters and determined whether or not that average frequency was increased or decreased in negative asymptomatic or symptomatic flu infection. So we have a lot of parameters. The Shivers group did a fantastic job collecting just tons and tons of data for us. So Amongst all of these covariates, we wanted to understand which were the most important in categorizing somebody, a participant in our study, as either asymptomatic or symptomatic. And what we did was use the decision tree or random forest analysis to start looking at our individual covariates. And what we did was we subsetted our data into two sets. So a training set, which allowed us to test our models, and then a validation set, or a testing set, which allowed us to test the models then on a second subset of the data. And so using a base model, which comprises just factors from these three groupings, we can see that our model does an okay job, about 60% accuracy of actually predicting or categorizing the participant as either symptomatic or asymptomatic for flu. But when we start building upon this base model to incorporate either the myeloid or the lymphoid or all of the components combined in this combined model, we can see that our accuracy improves to 84 and 86%. So just what this means is that just based on our baseline measurements or post-vax measurements, we can actually predict whether or not an individual, in this case, will potentially develop symptomatic flu or not during the course of the season. Pulling out from these models, what we can actually do is look at the variables that are most important in making those categorization determinations. And what you can appreciate, I think, is that the majority of these with the highest importance all come from our cell population compartments. And that's not to say that the demographic, serologic, and vaccine status aren't important, but in this particular case, the majority of those fall at the back end of the importance. So these decision and random forest modeling is great, but it actually doesn't tell us directionality. It just tells us the importance of an individual factor in making the categorization. That doesn't actually tell us whether or not you're at increased or reduced risk. And so to do that actual risk modeling, we did build a final logistic regression model. And so we did this two ways. We built one ourselves based on the information that we got from the univariate clustering and the variable of importance graph. We built our own model, and then we compared that using a stepwise AIC to a machine-built model. And the stepwise AIC takes all of those parameters and either adds individual parameters back or removes them to define a model that is the most accurate in predicting, in this case, risk of influenza, symptomatic influenza infection. And then what we did is we stress test both those models and compared them using Bayesian model averaging, and actually determined that our select model fared a bit better than the machine-generated one. And so the factor or the covariates in the final model comprise demographic and vaccine status, serology, and lymphoid and myeloid cell population covariates. And so I wish that the output of these logistic regressions were more exciting, but I have a nice table for you here. So the demographic and vaccine status parameters that were the most important in the model are shown here. Those serological factors that are most important are shown here, and lymphoid and myeloid cell populations under those. And what I've highlighted here are actually the individual covariates within the model that are significantly associated with either risk of susceptibility or reduced risk or protection during the course of the season. And I think interestingly, and historically, what we know is that that pre-existing antibody titers are kind of the classic hallmark of this is what we're going to measure for vaccine effectiveness, how well a vaccine is designed, and how protective it is. But in our case, we're not really seeing too many of these quote-unquote protective responses being significant predictors or identifiers of risk. And indeed, one factor that was significantly associated with susceptibility were pre-existing antibody titers, protective antibody titers to influenza B. We did see protective antibody titers against NAI titers against H3 being protective, which was really great. We know that this is likely amongst vaccinated individuals or individuals who had a prior exposure in the previous year. And then amongst the cell populations, nothing from the myeloid compartment came out, but we did see a number of lymphoid cell populations that were important in either increasing risk or allowing or affording protection. And several of these that increased susceptibility included CD4 effectors, CD8s producing a specific cytokine, TNF alpha, and inflammatory gamma delta T cells. And then interestingly, those that afforded protection were these CD107A or activated CD4 T cells, as well as naive and Th17 CD4s as well. So the CD4 populations were highly representative in our lymphoid compartment, potentially, especially with what we know about the relationship between CD4 T cells, subsets, and antibody production. This bodes well. And so what the last thing that we can actually do, because we have such great information about how these cell modules are actually co-correlated, we can do is use some of these cell populations that we know are strongly or significantly associated with protection or susceptibility risk. We can actually refer back to the various co-regulated immune cell module clusters. And then, although not all of these are represented in our final logistic regression model, we can actually use that information to kind of inform whether or not some of these other subpopulations might also be predictors of increased susceptibility or protection. And so, using the univariate and multivariate logistic regression modeling, we were able to build these protective and susceptibility cell profiles. One of the main takeaways is that while there are significant differences in the vaccinated and unvaccinated populations, there is a lot of overlap in which the vaccine populations kind of span the CD4 gambit. Unvaccinated are mostly what seem to be some sort of protective innate responses, and then the majority of those that are involved in susceptibility are inflammatory Th2 endothelial monocytes, gamma deltas, those types of things. So a lot of this is reflected in current literature as well. So where do we go from here? How are these conserved associations across diverse cohorts? And this is all from one area of one country. And we know through the CEIRR and CEIRR's network that we have access to all of these other different cohorts to not only look at different genetic backgrounds, different age groups, as well as test our models across different human cohorts. So with that, I would acknowledge my lab. Paul is a fantastic mentor. Everybody involved had a significant contribution to this study. I'll thank the study participants and specifically Sue Huang's group at Mission Risk 2 team and our funding sources from CEIRR. Thank you so much.
Okay, we can have one question. I'm going to pick Emily for a very quick one. So, very nice work. And you did a lot of work to try to balance all of these covariates in the model against each other when they're all sort of sometimes going to be having overlapping effects. It would be interesting to take a look and see, you could take, for instance, of those with high titers and low antibody titers and see what the marginal kind of effect of the cellular protection is on top of that and try to, now that you've aggregated it all, disaggregate some of these groups a little bit. I think that would be very interesting. I don't know if that's something that you've tried.
Yeah, we've done these analyses a couple of different types of ways, using either categorical versions of HAI or NAI, or to using continuous variables of them. But I'd love to chat with you afterwards and think about some additional ones too. Thank you.
Yes, sir. Okay. Thanks everyone for a stimulating discussion, and we're going to have a 10-minute break for a tea break. If you could come back at 11:05 for our second half of session 6, and encourage the questions at the panel discussion. Thanks.
Okay, so